Okay. Good afternoon, viewers at home. My name is Adeoti Olatunde Michael. This is another opportunity once again to continue in our educational series. And today we are focusing again on microbial evolution. Microbial evolution. And our topic is still microbial taxonomy and evolution of diversity. In this course, in the earlier video, we actually look at evolution as being a very powerful tool for speciation. Evolution is a powerful tool for speciation. And what do we mean by that? That is, there cannot be species if there is no evolution. The organism you see today, if you pick up any microorganism today or any organism at all, you just discover that that is the modern fashion of that microorganism, and that you need to trace it back evolutionarily to how it was in some years ago. And that is why there is no way we can do with classification or naming or assigning names to any organism if we are not actually relating it to where it is actually coming from in terms of evolution. So, and I told you in the last video that diversity actually means variability, different types of organism. Once you pick up any organism now, you just discover that that organism or the community of that organism contain a lot more of microorganisms which are related together and those that are not actually related together. That make it very possible to have range of different species of organisms with genetic variations and occupying different ecological niches in different ecosystems. That is talking about diversity. And again, when we now look at taxonomy, I told you the last time that the science of assigning names, describing organisms into their various taxa. I told you that taxon actually means group. That is, by the time you are grouping them, why are you grouping them? So that you'll be able to identify them, so that you'll be able to put them in the group that they actually belong to. And that is very important. And that is why taxonomy cannot be practiced without us knowing about systematics. And what is actually systematics? Systematics is the classification, identification, and naming of a microorganism or any organism based on their evolutionary relatedness. Based on their evolutionary relatedness. And another thing we actually look at on that day was ecotype. And I told you that ecotype is actually talking about members of microorganisms that they are evolution, they belong to the same, they are very related genetically. They are related genetically, but in terms of the ecology, their ecology, they are distinct from the same group, and that will now make you to be able to use differences in their ecology to be able to separate them. Although they may have almost similar genetic components or genetic contents, but because they are diverse, they are different in the area of their evolution. So that will make you to be able to separate them. And that is exactly what we mean if we are actually talking about ecotype. And the next other thing that we also look at that day was the melting temperature. I told you that this is the exact temperature at which the double-stranded DNA will begin to unwind and become separated at that certain temperature. So, and that temperature is actually known as TM temperature. So, and that is determined. This one is the melting temperature is determined by the amount or the number of their G plus C content. So, and in talking about G plus C content, I told you that this is the percentage in an organism genome, in the microbial genome, the percentage of guanine, adding it together with that of cytosine. Don't forget that according to the Chargaff rule, the number of guanine is going to be the same with that the number of cytosine that will be present. So it is based on this that we can also use the G plus C content, which will actually determine the melting temperature. So if you have G plus C content, if you have organism with high G plus C content, and you have other one with low G plus C content, that is telling you that the temperature at which their DNA will begin to unwind is going to be actually very, very different. And the next thing I actually mentioned was the RNA word. This is an hypothesis that actually believe that the first replicating entity was not DNA. They believe it was RNA because they said RNA is more stable than that of DNA. That DNA tends to undergo changes with time. Don't forget that I told you in our earlier video that microbiologists actually believe that our world is as old as 3.5 to 4.6 billion years ago. They now find out that there has been a kind of consistency in the component constitution and consistency of the RNAs. So RNA was more stable, and that was why they so much believe that RNA was the first dividing entity that you can use in classifying them. And that was one of the reason why Carl Schweitzer, who actually divided the organism into 3 domains of life. All the living organisms were classified into 3 based on their SSU, ribosomal subunit. We are going to see more of this as we go on. And this is just a kind of revision of what we have said in the earlier video. And again, I also mentioned something about universal phylogenetic tree. So this was a belief that considered that evolutionary relationship among organisms that they were able to use this phylogenetic tree to classify organisms into 3 domains of life. That is archaea, eukarya, and prokarya. So they actually make use of a universal phylogenetic tree where these 3 domains had their origin, their roots from the same origin, and that is what is known as a universal phylogenetic tree. And after that, we now look at classification of microorganism based on classification. So there are different types of classification that have been used in classifying organisms into their various groups. But the one that is as old as Adam and Eve was the use of physical physical characterization, just like a possession of flagella, their morphological structure, to be able to classify microorganisms into their various taxa. And by the time you are making use of this, you just discover that erroneously you are going to place E. coli and Toxoplasma gondii, which is a parasite. You are going to place them together because two of them have a flagella, and by making use of that, you are going to make error in classification. And that was when and why scientists actually moved from the era of using phonetic classification into the era of believing in phylogenetic classification. The phylogenetic classification actually believe that organisms, they have a very common root, and that as a result of their evolutionary relatedness, that is exactly what is going to place them in together. That is, if they have similar evolutionary relatedness, they will be placed together, and those ones that are having distant relationship, they will be placed in a very different group. And this was the classification that was made by Carl Woese, who actually used ribosomal SSU ribosomal RNA in classifying microorganisms. So it was based on their evolutionary trees, and it was based that ribosomes are more stable. Ribosomes are more stable, and the scientists could actually rely on them for classification of organisms, especially microorganisms. So, and that was exactly what Charles Darwin believed in when he published his book on the origin of species, and that was exactly what actually happened. So the next type of classification after phylogenetic classification was genotypic classification. This was a kind of classification that actually was based on their genotype. So microorganisms that have very closely related genotype, they are bound to be related, and those ones that are distantly related in terms of their genotypic constitution will actually have them to be grouped in a very different group. So, and that was that of classification. And now the question now is why? Why do we actually classify? Why? Why do we actually classify? Why do we actually practice systematics? Why is it very possible for us to be able to identify, to categorize, to classify them based on their taxonomic hierarchy? So, and when we now talk of hierarchy actually means that you are moving from millions. You are moving, when you consider millions under the, okay, and the taxonomic hierarchy that is very, very applicable in microbiology is the one that has the first one, that is domain. The first one, which contains billions of microorganisms that will occupy the same domain. So when Carl Woese grouped all the organisms into 3 domains, that means there are some organisms that you found them in the same domain, but they are distantly related. But in terms of their ribosomal RNA, they are grouped together. So, and that's why the first taxonomic hierarchy in microbiology is domain, then followed by kingdom, followed by phylum, followed by the class, followed by order, followed by family, followed by genus, followed by species and sub-species. Don't forget that in the earlier video, I defined species for you as the group of related microorganisms or organisms that are capable of interbreeding together. That is, they can reproduce together. So that is the lowest taxonomic hierarchy. Now the next thing is now to look at what are the techniques that you as a microbiologist, what are those techniques that you can actually use to be able to classify microorganisms. To be able to classify microorganisms, don't forget that why do we actually have to give names or assign names to microorganisms is to be able to identify them, is be able to know them, is so for you to be able to diagnose, and you should be able to, when you diagnose them correctly, that means you should be able to also give them very good and correct treatment. So as a microbiologist today, you should arm yourself with different methods that you can use in classifying them. I've actually warned you that gone were those days that as a microbiologist you just depend on the use of cultural method to be able to classify your microorganisms or this, and at the time you now use cultural method, you just come and do prescription based on that. If you are doing prescription based on that, that means you are misdiagnosing and you are being tempted to be able to mistreat as well. And when you mistreat, you know you are treating and teaching the microorganism to become more resistant. And that's one of the reasons why abuse and misuse of drugs is still a very powerful factor, that we now have resistant strains or resistant species of microorganisms. So these are basic techniques that we can use in medical diagnosis of or clinical diagnosis of some ailment. So don't forget that I told you that sometimes you might grow your culture, you might grow microorganism on plate, and at the end of the day, when you grow them on plate, you just discover that although morphologically they have the same, maybe their whitish, their colonial appearance is very the same and similar, and by the time you now go on, you just go and call them. For example, now part of classifying or identification of a microorganism, some hospitals, some people still believe in what is called presumptive diagnosis. Presumptive diagnosis is a diagnosis that is based on symptoms. You just discover that, oh, once you have severe headache, oh, you are suffering from typhoid. No, because there is not too many or too much differences between the symptom that is being put forward by Plasmodium species, which is the causative agent of malaria, and the one that will also be put up by Salmonella typhi or Shigella sometimes, because these three could actually come with very similar symptoms. So when you now depend on presumptive diagnosis, you are bound to misdiagnose as well. So the first techniques that you can use, that is the lowest level that you can start your diagnosis. The first one is the one that is based on classical characteristics. What are classical characteristics? These are characteristics that is natural, that anybody can actually see, that when you, in fact, you don't even actually need more training. You don't actually need to train your hand before you be able to see this is exactly what is actually going on. So classical means that is a type of diagnosis that anybody at all, the person might not actually need to be a professional. before they will be able to use that, and that is classical. That is the one that is natural, the one that is another word for classical is traditional. It's traditional, it's cultural, the one that everybody has got to inherit from our forefathers. That is classical, and that is the classification that is based on morphological appearance. Maybe the organism has flagella or it doesn't, colonial appearance. So these are parts of what is actually based on that. You can also base it on sizes, you can base it on structure. So that time I made mention of some of these, which include the cell shape. If you see them under the microscope, how do they look like? That's you say, oh, the one that is Staphylococcus will occur in this form, and the one that is Streptococcus is going to occur in chains. That of Staphylococcus is going to be in bundles. So all these are part of a classical identification or classical classification of microorganisms. Then you can also use, apart from the cell shape, you use cell size, you use colonial morphology, you can use their staining technique, whether they are able to stain positive with gram or they are staining negative with gram, whether they have cilia or they have flagella. What is the mechanism they use for motility? Is it grinding, that you classify them into grinding bacteria, or are you going to classify them according to the way spirochetes are being classified? And sometimes you can also use endospore shape, whether they have, what is the shape of their endospore? Do they have endospore? Do they undergo spores or spore forming? So that will actually make you to be able to separate a bacterial spore. Bacterial spore is quite different from fungal spores. The way they appear is quite different, and even protists as well. So be able to actually differentiate them into that. And there are some other bacteria or other microbes that they also have some other cellular inclusion, apart from the fact that they have DNA, apart from the fact that they have ribosomes, apart from the fact that some of them have pili. There are some that still have other cellular inclusions, which their functions might not actually be known. Then classification that is also based on color. Classification that is based on color is also part of classical classification. And the one that is also based on the second one, which is a physiological stroke metabolic classification, is the one that is based on where do they get their carbon and nitrogen sources? Where are they getting it from? It could be microorganism could be classified based on the source of carbon and the source of their nitrogen using metabolic classification. Then some could be also classified in terms of energy. Where do they get their energy from? For example, those ones that obtain their energy from the sun, you classify them as photosynthetic. And those ones that we actually derive their energy from chemicals, you classify them as a chemosynthetic, and so on. Then sometimes physiological is also the one that you use their nutritional type to classify them. Are they lactose fermenters or non-lactose fermenter? You also use growth temperature. At what temperature do they grow? Most. That's where you classify some as a mesophilic, you classify them as thermophilic, and you classify them as a thermophile. Then you also classify them based on their osmotic tolerance, their ability to be able to tolerate some pressures. Their pH optimum can also be used. the presence or absence of photosynthetic pigment. You can also use this in classifying them. Also, you can also use their salt requirements and tolerance to be able to classify. All these are falling under physiological stroke metabolic classification. So, and sometimes after that, we can also classify them based on some chemicals. For example, those ones that have enzymes like catalase. That is, they have catalase that can actually break down hydrogen peroxide, utilize hydrogen peroxide into water and oxygen. You classify them as a catalase positive and so on. And there are some that use cultural method of classifying them. But you see, once you believe in that, oh, let me just culture them and let me see how it goes, you will know. For example, when you are using nutritional growth to classify them, you should know that the way, if you want to culture Mycobacterium tuberculosis, that is the causative agent of tuberculosis. Now, you do not use all these common agar in classifying them. You don't use maybe potato dextrose. You don't use that. You don't use McConkey. You don't use chocolate. You don't use all this. They have their own special agar they use in culturing them, and that agar must be egg based. For example, the solid agar is a Lowenstein-Jensen agar, and there are some microorganisms that are non-culturable. You cannot actually culture them in the lab. You cannot culture them in the lab. So, and that is exactly what you should know. One of the errors of basing your classification based on physiological or metabolic classification. Then sometimes when you now use ecological classification, that is where you find them, now you depend on that in order to be able to classify them. You just discover that in those days, you want to classify E. coli as enteric, that is a bacteria that you found them in the guts. But nowadays, you just discover that you found them in the environment, you find some of them in water, you find them almost every ecological niches. So by the time you now do that, and what you believe in is that you are just trying to believe in rely on their ecological distinct in classifying them, that means you are going to make a lot of errors. So, and that's the reason why some of the defects or some of the demerits or weaknesses of a classical classification, which I call traditional method of classification. And now, scientists have now discovered that making use of that have actually made them to be able to misdiagnose some of them. Some of them could not be cultured. Some of them you cannot even see them without a specialty. So that was why scientists now came up with the most advanced now, which is molecular. And that molecular, they started it with in terms of nucleic acid based constitution. Nucleic acid based constitution. I told you earlier on that microorganisms will also be classified based on their G+C contents. So, and if you want to know that, I've given you that formula. I also gave you this formula the last time, that if you want to know the molecular, you want to know the GC content of any microorganism or any microorganism, what you do is you will have a G+C all over, that is the percentage of G+C is equal to G+C all over G+C plus A plus T. Then you multiply it by 100%. So that is going to give you the amount of G+C content. And that amount will determine the melting temperature. That is the temperature at which the DNA strand of the organism into that in that group will now melt. So they will now become unwinded. So, and we have diverse microorganisms that have been classified as low G plus C content, and those ones that have been known to be high G plus C content. We are going to pick this. We are going to dedicate some series of lectures for considering what are the basic characteristics, what are the basic components of bacteria or microorganism that belong to G plus C, low G plus C, and what are those ones that you can also call high G plus C. We are going to see all this as we go on. Then the next one is the use of nucleic acid sequencing. Nucleic acid sequencing is one of the most ever reliable, very efficient, and very specific. So you use their nucleic acid, like, and when you talk of nucleic acid sequences, you are sequencing techniques, you are actually referring to DNA or RNA sequencing. For example, well, that's when you talk of nucleic acid hybridization, that is DNA to DNA hybridization, and the RNA, that is, you can have it RNA to DNA hybridization technique. You can also have that as well. So you have a lot under nucleic acid sequencing, and we are going to consider at any point in time, we are going to consider whole genome sequencing techniques, as well as partial sequencing techniques. So we are going to see all this. Then the last one that we did not mention, that time is a genomic fingerprinting. So let me now start today with a genomic fingerprinting. Don't forget that all what we have been saying so far behind the camera, these are part of a kind of revision into what I've said so far concerning microbial ecology and diversity. I told you that diversity, there is power, there is strength in diversity. Just like Nigeria is made up of different tribes, and it is that diversity that is making us to stand together. The same thing here, the same thing in microorganism is that there is strength in their diversity. When you pick a community of bacteria, if you pick a community of microorganisms now, you'll discover that in that same small community, you can have more than millions species of microorganisms. You can have related and those ones that are unrelated. And don't forget that I told you that the practice of identification, the practice of naming, and the practice of grouping of these microorganisms into their various groups is exactly what we are actually interested in medical or clinical microbiology. Because don't forget that I told you the primary aim of identification, the primary aim of naming, because everybody has his own name, and just like they normally say in our culture, that because somebody is going to commit a crime, that is why everybody is being assigned a different name. So, and everybody has gotten his own identity by their names. So in a class of 500 now, you just discover that you have people that will never bear the same surname, and there are some that they bear the same surname and they are not related at all. But once you are able to identify them based on their name, that one is going to help you for identification, and that is what is our primary focus in medical diagnosis or medical microbiology. So just like I said, we have, okay, all this one, every other thing now, we have treated them in some of in the earlier videos, and that is why today we only want to make mention of genomic fingerprinting. Genomic fingerprinting. Oh, when we say genomic fingerprinting, it is just that we believe that microorganisms, if you want to identify microorganisms now, you need to go down into the gene level, genetic level, genomic level. Not only one gene, you should be able to harvest that microorganism and harvest it in form of that you have its genome. That is the total sum of all the genes in any microorganism. And don't forget that genomic fingerprinting, you know, we believe that fingerprint, if you want to identify anybody that has committed any crime, so they will ask you to go and thumbprint. So the same thing with genomic fingerprinting. The same thing is that at genomic level, every microorganism has gotten a different fingerprinting, a different genetic identity, genomic identity at the genomic fingerprinting. And that is why we actually have to be very, very what? Be very, very specific. And that is the evaluation. That is the evaluation. Genomic fingerprinting is the evaluation that believes or that rely on that every microorganism at the genomic level will have different types of gene that will code for their transfer RNA. So that is at a genomic level, and that's why they also believe in what a process that we call it RFLP. RFLP, that is the enzymes. That is at the enzymatic level that they believe so much that you cannot, which is called restriction fragment length polymorphism. So this is restriction fragment length polymorphism. This belief that each microorganism or each organism will have a unique identity of how they can break down enzymes into different patterns. For example, now, if you ask some people, let's say you have different loaves of bread, and you will give so many people, no matter the type of training you might subject them to, you not give them knife that they should slice that loaf of bread. So, and you give them knife, you will just discover that each of them is going to slice that loaf of bread in a very unique pattern that is very unique to their own identity. So the same thing with scientists, which is called genomic fingerprinting. So that is the level that, for example, now let's now consider a typical example of how that is hypothetical example of how you can make use of genomic fingerprinting in diagnosis of a very simple infection. For example, now let's say at this level, let's say you collect sample, and don't forget that when you collect sample, before you collect sample, there are some factors that would determine the type of sample you will collect, how you will collect it, and how it is going to be transported. So something must have guided you. Something must have guided you. The organism you are going to get from blood sample is going to be quite different from the one you get from sputum. It's going to be quite different from the one you get from the skin, swab, and so on and so forth. So that means at that point in time, you must have actually gotten some certain intuitions or training that will direct you on what to collect. So, and by the time you collect the sample now, you collect that sample, okay? By the time you collect the sample, so the sample could be from swab, could be swab, could be blood, could be anything. So by the time you collect that, and the way you also transport it also matters, and some of the precautions that you take also matters, so that at the end of the day you are going to have a lot of this. So the next thing you do is to go and culture. You use culture either by serial dilution, and at this point in time, you must have been guided by different types of agar that you are going to use. Before you determine the agar you are going to use, that means you must have gotten you must have been familiar with the types of nutrition, the type of agar that is going to support the growth of the will-be microorganism that you are about to diagnose. That is, we want to look at typical examples of how you can use genomic fingerprinting in diagnosis or in clinical diagnosis of a certain specific infectious agents. I told you collect samples, and that after collection of that samples, you will get it inside the bottle. So, and the type of sample will also determine the type of bottle you are going to use. If you are going to collect sputum now, you are not going to use this. If you are going to collect blood, that is when you may use something like this. You use EDTA, some that will not contain anticoagulant, and all this one, they are part of what should have guided you. So at that point in time, the next thing is now to take this into the laboratory. And by the time you are taking it into the laboratory, transportation, the way you transport it, the way you store it also matter very, very well. So you take it to that level, and at that point in time, you will now determine the type of culture or the type of agar you are going to use. Are you going to use solid agar? Is it going to be solid? Is it going to be semi-solid? Is it going to be liquid? So all this matters. All this matters a lot. So, and if you are talking about solid agar, you have the one that are general purpose, you have the one that are selective, you have some that are differential, and some that are enriched and enrichment. So all this will guide you at that culture level. Now the question is, now this is where the problem lies. You will only proceed, that is, if the microorganism that is causing the infection that you want to actually diagnose can be cultured in the lab. If it cannot be cultured, you know, I said it behind the camera the other time, that there are some microorganisms that are non-culturable. They are not culturable, and that will leave us with another different thing entirely. But let's consider those ones that are culturable, and that is when you can make use of this. So when you now culture, immediately you culture, you introduce a lot of biochemical, physiological, all this one. So the next thing is to get the DNA. To get the DNA by the use of DNA extraction principle. You should know at least at certain point in time, you will get DNA. You will extract their DNA, and by the time you now extract their DNA, what do you want to do with their DNA? At the DNA level, you are already entering into the molecular. So from here to this point, you are still making use of a classical method. Are still making use of classical methods. So classical methods is what you can make use of. So sometimes between here and before you get to the DNA level, you could also make use of serological methods. Serological methods. So, such as the one you use ELISA technique. For example, if you are dealing with HIV, if it is a virus now that you want to diagnose, that means you collect sample. After the sample, whatever you must have done, you will do RDT, that is rapid diagnostic test, by the use of strips. So you need to do confirmatory test by the use of serological, that is antigen antibody reaction binding pattern. So that is serological. So this will come as in between, that is number 2, which I did not mention before. This is going to be number 2. Then from this DNA level, you now start what is called molecular techniques. Okay, so at that molecular techniques, so you can make use of what is some of the method that you can use is called DNA hybridization technique. You can use the G+C contents, just like I said the other one, DNA hybridization. You can now use so after that, you now use you can use PCR. This PCR you just get a small fragment of your DNA, just small fragment, and you magnify it. So that is a polymerase chain reaction. You magnify it. That is called amplification. So you must have amplified your DNA, very small fragment. It could be because one of the advantages of molecular diagnosis, is that the causative agents of some certain diseases might not actually be what you won't be able to. Okay, if the causative agent is DNA, you just discover that by the time you are culturing the DNA, you can only culture for an organism that is still alive, that is still living. If it is no longer living, it's not going to grow at the end of the day. So that is the reason why, even when you must have finished your classical method, you need to subject it into molecular, because at that molecular level, even if you pick if the problem is going to be from a very small fragment of that DNA, you will be able to pick it at molecular level, and that DNA is going to be amplified through the process which is called PCR. And there are different types of PCR. We have nested, we have single SSA, we have a lot of PCR. But what they do is just that what PCR does is that it will amplify. That's why they call it polymerase chain reaction. So it's going to amplify at that level. Now, after that amplification, we are going to now use electrophoresis technique. That is the technique that we are now calling what? Electrophoresis tank. That is where you make use of your RFLP method at electrophoresis. So that is when you'll be able to observe, the traveling pattern and the ionic pattern of the DNA. Yes, that is ability to move in what? Ability to, okay, that's going to be like this. So this is going to be a gel, and this one and this one, okay, you could have different wells. So the first one is going to be control, or what you can call ladder, leading ladder. So you drop your DNA sample here, sample 1. Let's say you drop one here, you drop another one here, and you will now place this one in electrophoretic tank, and you now observe it, you leave it and pass electric current over it. So by the time electric current travels over all these amplified samples, it's going to come up with a different pattern. The DNA, you will see it according to the size, or sizes. So it's going to be like this. It's going to be, so it's going to be like this. So this is well A, this is B, this is control, this is 1. For example, let's say this is 2. This is going to be the control or the ladder. So the DNA here, by the time you subject this to electrical current, is going to travel to the, based on the weights. The one that is heaviest, will not travel too far from the well, and the one that is the lightest will be the one that will travel far. So it is at that point that you call it so after this, based on their RFLP pattern, that is the way this one is going to settle out. So this one will now be eluted. You now elute this DNA, and by the time you elute that DNA, you now take it into you take it for sequencing. You take it for what is called sequencing. And don't forget that I told you that we have whole genome sequencing, whole genome, that is the totality of that gene. Whole genome sequencing is going to predict the total gene in that organism. And we also have partial sequencing, like the one you use Sanger principle for, and the whole genome, this one. We're going to see all this. But at whole genome sequencing, that is when you now have what is called genomic fingerprinting. You have what is called genomic fingerprinting. And it is, look at where you have started your diagnosis. You have started from collection of samples, and look at what you have subjected it to. Before it is going to get to this level, at least you must have known, you must have been certain that, or identify, you must have been able to identify that, oh, this is the exact problem. This is the exact cause of that problem. So at that whole genome sequencing, you are still not going to stop at that point. So the next thing you will do after whole genome sequencing is to now collect the sequences. You collect the sequences now, which is going to be in form of G, C, A, T, C, A, T, blah blah, like that. So by the time you now do that, you will collect that. You now use what is called bioinformatics tools. Bioinformatic tools to be able to identify. You now use bioinformatics tools to be able to identify. That is when you now collect your results in form of sequences. and the next thing is to feed it into the computer. And from that computer, you can do a lot of other manipulation that you will now be able to identify. You will now be so specific. Don't forget some of the application of bioinformatics tools is individual or what is called personalized medicine this time around. So all this information, by the time you have inputted it into your computer, and you are able to use appropriate bioinformatics tool to be able to interpret and manipulate this, that one is now going to guide a clinician or a medical doctor that will be able to prescribe drugs that is going to be very specific for you. In fact, at bioinformatics level, we now use what we call drug design techniques, that even at that point in time, they will be able to design drugs for you, that they will just take that sequences, they will take it into the lab, and that drug that will be produced for you is going to be only and only meant for you and no other person. So that is at that point in time, look at where we're actually starting from. So at this point that you are so sure that whatever they must have identified as the problem, it is exactly what is done. So that is that about genomic fingerprinting. And also the same thing also follows at amino acid sequences. So don't forget that at the end of the sequencing, they will take it again they will subject it again to amino acid. Don't forget what I said, that you started from DNA, RNA, and you have amino acids, different amino acid which is forming all this. So that means you are not stopping your diagnosis at DNA level. You are stopping your diagnosis at amino acid level, and that is what's going to also inform you carrying out using spectrophotometer, whereby that is actually meant for diagnosis of protein, colorimeter, and so on, that you can use it in amino acid. And this time around, you are sequencing the whole amino acid, and the practice of that is no longer called genomics. It is called proteomics, because we are going to be talking in amino acids. Don't forget that we have 3 triplet codon. 3 of these is going to code for an amino acid. So it is at that level that you are actually dealing with, and that is going to be the last of your diagnosis. So you can actually see the way and the technique, the pattern that we can now use in classification of microorganisms. So, and any classification that you do at this level, is going to be more accurate and more appropriate. So that is what you also need to know at this point in time in talking about the evolution of taxonomy and the classification of microorganisms based on talk about classical pattern, classical method, which is traditional, then followed by serological, that is actually based on antigen antibody behavior reaction. Then before you come into molecular, it's going to lead you to genomic as a proteomics level. And it is at that point in time that scientists nowadays now start looking at bioinformatics tools. That is why microbiology today is not complete if you do not have knowledge in bioinformatics and how it can be applied in drug design, that is pharmaceutical industries, how it can be used in vaccine production, and how it can also be used in metabolomics, that is predicting some of the metabolic pathways of a microorganism. So, and that is the what we need to do, and that's just the trend in clinical diagnosis of major infection, and which you must know. Don't forget that this is a digital world, and everything you are going to do, you are going to rely on data, and that is where we have gotten to, where you have everything that started from collection of that sample. You have now digitalized it at bioinformatics level. So don't forget to click notification button, so that anytime we drop any video, you'll be able to be notified. Like, subscribe. Thank you.